Good morning to all of you. So today we are pleased to welcome Nicolas Schtickzelle. No, it's not correct. Maybe you can correct me.
Schtickzelle.
Okay, thank you.
You passed, but just.
Okay, thanks. And so Nicolas is in Toulouse for 2 days or 3 days. He's invited in the context of the graduate school, and because he participated to the formation of our Master 2 student. And thanks for coming for that and for giving some courses and for the student. And we have another seminar today because we have another guest in Australia. We will give also a seminar tomorrow, and Mathieu Joron also on Friday. So if people want to discuss with Mathieu, he will be free tomorrow afternoon. Okay, concerning Nicolas, a few words about you, really few words, because it will take a lot of time and it's better to keep time for your talk. So you are professor of quantitative ecology and conservation biology, right? At the University of Catholic in Louvain in Belgium. And so you are interested by how population can react to global changes such as pollution, climate change, and many other constraints, I suppose, and how this constraint can affect the variability of this population. So I prefer to stop here and to let you present your seminar, and thanks a lot for coming.
Thanks for inviting me. It's a pleasure to be here and to talk about results on dispersal on microcosm. So I started my career working on threatened butterflies in the field. I was mainly interested in the effect of habitat fragmentation and understanding how metapopulation develop in the field. And then after I had the chance to go for postdoc in Paris with Jean Claubert, who's now in Moulis, I was supposed to do some quite complicated capture-recapture data analysis on my butterfly data. And when I arrived, Jean told me, I have something else that you're likely to be very happy to work on. And basically, this is the start of the Tetramina thermophila microcosms. And sure, I'm presenting this synthesis today, but this is the work of many collaborators. And this collaboration between me and now my team and Jean Claubert and all the other researchers now in Moulis is still very active, and you will recognize a few names in there. So I call that dispersal in a bottle in artificial microcosms, and we're going to speak about drivers and also intraspecific variation in movement, but in dispersal too. So let's start with a brief recap on what is dispersal. So dispersal is not any movement, okay? Dispersal is a movement where one individual will end up in another population, potentially reproducing. So dispersal is a movement which can potentially lead to gene flow. Why potentially? Well, this has been a struggle in the early years between people with a more genetic view and those with a demographic view, whether if an individual moves from one population to another but fails to reproduce, is it dispersal? Okay. Well, with a demographic background, I always tended to say yes. Why? Because just imagine you're a butterfly, you are a female butterfly, and you have few males that comes. In this species, the butterfly, females mate once and male mate many times. Okay. So females' fitness is going to be negatively affected by the density of males, because females, once mated, they are going to be harassed by males, and they will lose opportunity to select good places to lay eggs. Okay? So a male who comes and harasses females will have an impact on the demography of this population, even if it fails to reproduce. Dispersal is not migration. So in the literature, there has been many cases where the two terms have been used interchangeably. especially because in the population genetic background, there was a tendency to use migration to basically, migrants are individuals that create gene flow, okay? But we should try to keep migration for what it is. So migration is a periodic back and forth movement between two different habitats due to the life cycle. Storks migrates, okay? Toads also. Toads will reproduce in a pond and will live the other of the rest of the life cycle in the forest. Okay, so it can be very close, but this is obligatory back and forth. Just imagine movement of salmon population. This is the circile salmon you can find in the Pacific Northwest. This is an obligatory migratory. So fish, they are born in rivers and they need to go to the sea to just grow for a few years. So all individuals of this species must go to sea and then they come back. Okay, so this can be thousands of kilometers. Just take that salmon, it's going to sea, and then it's coming back in the same place. It moved a lot. It did a migration event, but it did not disperse because this individual will end up reproducing at the same exact place where it was born. But if it comes back to another population, obviously there is a migration event, but there is also a dispersal. Okay, so I'm not interested in migration, but I think this was important to put. So, well, you will still find a lot of use of migrants, the number of migrants per generation, and so on. Well, we should try step by step to replace that in our use. If you mean a movement from the natal population to another population, let's call that dispersal. So this is a very specific type of movement. And all species disperse because on the very long term, without dispersal, the fate is extinction. I mean, it can be very, very long time, but you need somehow to be able to colonize new areas to adapt to the changes in your environment. Okay, it could be active, like butterflies that's going to fly. It could also be completely passive. Okay, plants disperse passively. All this is basically the way they will disperse. The timing also can be very variable. It doesn't matter. Okay, it's still dispersal events. And also, dispersal can occur only one time. For plants, it's usually the case. I mean, the seed can just be flown away and then germinate, while the butterflies I was studying some individuals will go and reproduce in many different populations. And dispersal is really a key ecological process because, well, depending at which spatial scale or process you look, okay, it is what allows recognition of habitats where population disappeared. So this is more in a kind of population or metapopulation dynamics. Okay, so dispersal is what allows metapopulation dynamics. So I'm not sure you're familiar with what is a metapopulation, but in a nutshell, a metapopulation is a population of populations. Okay, so what is a population? It's just a set of individuals. And why the population can have a lifetime much longer than any individual is because individuals are born and die asynchronously. Okay? So not everyone die at the same time, and then you can have the population will stay quite a long time. And metapopulation is exactly the same. Populations will go extinct. New population will be recognized by dispersal. And provided all these extinctions do not occur at the same time, a metapopulation can have a very long lifetime. Okay? Obviously, dispersal is also what allows range shifts in the case of climate change. And without speaking about everything linked to gene flow, adaptation, evolution. Okay. So really, dispersal is a key ecological process. For a very long time, and when I was a young student working on butterflies, you could find books, of butterflies, like lists of butterflies, and species were labeled as sedentary or mobile. So the view was, dispersal is a fixed species attribute. Some species disperse, some not. And there was a kind of a confusion also between dispersal being not all movements. Okay, so the monarch butterfly is in the US is migrating. Okay, I'm sure it was cataloged as mobile, but it does not mean anything about dispersal. And Jean Clauber had a big impact in the fields of dispersal ecology. More and more evidence show that, in fact, dispersal is much more complex than that. It's not fixed, and it's not purely random. Dispersal is context dependent. This is an example of a study I made during my PhD thesis, is comparing the dispersal of this bog fritillary butterfly in 4 different landscapes with different habitat fragmentation. So these 3 are in Belgium. Here you can barely see the habitat patches, okay? And this is in Finland, where the habitat is globally continuous. And just one result we got comparing these. This is, so you get these 4 different landscapes from the less fragmented to the more fragmented. And just look at the black dots, okay? This is the standardized emigration rate. So, while the same individuals, well, they are not the same individuals in a way that maybe they are adapted differently, but the same species, okay, will have a tendency to restrict from emigrating in a landscape like this, where the cost of dispersal is going to be much bigger. Because what is the likelihood that if a butterfly decides to emigrate from that patch, what is the likelihood that he will end up alive and not exhausted into another one? So it's context dependent. It's also condition dependent. It depends on how the individual perceives its condition in the context. This is just one of the quite easy example where species have different morphs. Okay, this is fixed for life, but we will see with the Tetramena microcosm, the species, that it can also not be fixed for life as a developmental features, but individual with wings are going to be the dispersal individuals and not those. So understanding dispersal is quite important if we want to better understand eco-evolutionary dynamics, and even further in a global change context, where dispersal will be a key process to help species face with global change. Because when your environmental change, either you die, or you adapt, or you move to find something good elsewhere, okay? And adaptation may not be so fast. So I told you I started my career and I still continue working on butterflies. That's great. That's where, as a biologist, a conservation biologist, so my aim as a scientist is to create scientific knowledge to help conserve species. Obviously, it's in nature that the real thing is happening. But this butterfly is a threatened species, it's a protected species, and there are a lot of limitations working with that. Once I got a comment on a paper I sent for publication, very nice sentence. Oh, your study is really great, but come back when you have 50 populations more. Nice, okay? So there are a lot of limitations. So I had the chance, it's maybe a chance of even, it's just Jean Clauber who told me, okay, maybe you can try that. It was totally not planned, but in a very well, that I started the second complementary research axis on laboratory microcosms. So on the field, you get the realism. In microcosms, or mesocosm or whatever, more controlled systems, you trade off this realism for control, manipulation, and replication. Okay? And the idea of developing ciliated protists, Tetramena thermophila, that's the species, I'll tell you that later. The idea of using that species for ecology is Jean-Claude's idea. This species has been used in molecular biology for decades and decades, okay? But he had the idea that maybe it's a good model to study this person. So why these ciliated protist microcosms are basically a good system to study this person? Because it's not so obvious. I mean, talk to any, I would say, standard ecologist, okay, especially if you lean towards conservation biology, bottle or lab ecology. No, that's not real. Okay, it's a very crazy idea. But they have a lot of advantages. So in microcosm, as I said, you have a high level of control and replication, and you can manipulate a lot of things. Just to example, you can manipulate landscape configuration. So this is an example of a simplified two-patch system. It's just two appenders, two microtubes connected with a silicon pipe. Okay. These are more recent 3D printed landscape. This is something I have developed for years and years. It's still not perfectly working because on paper it's extremely easy, but in fact, technical details are always, the devil lies in the details, you know that. But still we can manipulate that, something that would never be able to do with butterflies. Interested by climate change? Well, it's so easy to just change the temperature of the incubator in which you put that. So that's for the microcosm. And why ciliated protists? These start now to really be established models in ecology and evolution. And especially for dispersal, one of the big interests is that they are actively dispersing. Okay? So this means you can study dispersal in these simplified systems. Okay? Dispersal, that is the consequence of individual choice and individual movement. Okay? I have colleagues working on microcosm with algae. They are not interested in dispersal. I mean, it will be totally useless because they will just drop at the bottom of the tank. That's it. Okay. These species, they do sexual reproduction, but they mainly do clonal reproduction. So this means you can just get millions and millions of individuals that are all genetically similar. So we can have emitting in lab several genotypes, several strains. And these strains, this is the Tetramena thermophila, so one of the species that is most used in these ciliated protist microcosm. It's the main in which Mullis and we work. We both have different species, but that's the core species. And these strains, they exhibit very different phenotypes and life history traits. And this is, for example, what is of interest to me in a conservation context, because what is something is very important in conservation? To be able to predict the reaction of species, for example, habitat fragmentation, according to the life history traits. It's assumed that a species that will be a better disperser would better cope with habitat fragmentation. But it's quite hard to test in nature. While here, we have different strains that disperse more or less. Okay? Sure, they are not different species, but what is a species? It's a collection of individuals that are closer, more similar together than others. One strain of Tetramena compared to another strain. All the individuals within a strain, they are quite similar and much more similar than another one. So this is an analogy that allows to study some questions I'm interested in for conservation biology, even if it's in artificial systems. And this system is especially good to perform G by E experiments, so genotype by environment, to see what is the impact on the intraspecific variability. Okay, whoops, not easy with this. It's disappearing. Okay, or the G. Oh, what is the impact of the environment? So the E, the environment, allows you to study phenotypic plasticity, which is the fact that the same genotype will produce different phenotypes in different environments. And the interaction. And what is the G by E? It's the variation between different genotypes, the G, in their plasticity to environment. So this is the evolutionary potential for adaptation to the reaction to this environment. And this species, this Tetramenous Haemophila, also can create a dispersal morph. So under specific condition of food starvation, cells are going to transform themselves, elongate, and create a flagellum. And this allows them to be long-distance dispersal morphs. In this video, it works because it's on a, okay, I had to move that to a Mac, and this, okay. You should have been able to see that some individuals, all move relatively, tortuously and so on, and some just cross the screen fast and straight, okay? So over the years, with some student of mine or with the people in Maurice and so on, we basically developed different techniques of measuring abundance, life history traits, and movement traits using image analysis. Okay? So we get abundance, morphology, and we are able to automatically trace the trajectories. So it allows to record a lot of information relatively easily. I'm not going to do it. Okay, so the first thing we observed is that strains differ in nearly everything. Each time we look at the trait, there is a relatively large variation. Here I'm focusing on dispersal. And in very simple two-patch system like that, so all is filled with medium. So we can discuss whether this is a real dispersal corridor or not, because a corridor should not be suitable for other habitat. But okay, that's a simplification of the system. You put the cell in one tube, you let them disperse for a few hours. At the end, you kill them, and you look how many moved, and you can measure the traits, the movement, morphology of the residents and the dispersals. So first is that some strains, so here the strains, we have 44, okay? Some come directly from nature, some are inbred in the labs, inbred of, okay? So this makes a quite interesting variation in terms of genetic background. Some have a very low dispersal rate, and some have a very high dispersal rate. And it's relatively repeatable. You see here, you have 3 replicates, and this is quite repeatable. Also, the movement behavior differs. So some swim fast, some swim very straight, and some are slow and tortuous. Okay. More interesting, if you look, for example, this speed graph here, okay, one point and line is one strain, so you see the strain difference here. But the line connects the residents and the dispersers of the same strain. So interestingly, you also see a huge variation that the movement, there are variation between dispersers and residents, and it's not always in the same direction. And this is also quite repeatable. So it's just not random noise. So we try to put both together. Is in such a system, is dispersal just a byproduct of movement? Maybe it's just foraging movement. The more they move, the more they disperse. Okay. Simple model, we can predict 50%. So we can predict 50% of this variation by taking into account the variation in movement. So it's a bottle half full, half empty. Okay. So we also relatively rapidly observed that dispersal, all traits are correlated. And especially dispersals are correlated to other traits in what is called dispersal syndromes. Okay, just to give you one example, this is one of one of the very early studies I did with Jean in Paris, only on a subset of, we only had 10 strains at the beginning. And one quite clear example, so this is a PCA, so it shows how different traits are correlated. And here dispersal, so strains that tend to disperse a lot tend to be less cooperative, okay? But strains that are cooperative here do not disperse, but make a long number of dispersal modes. And this makes sense, okay? If you are cooperative, why would the benefit of kin cooperation? That's because you favor your kin. So you should stay with your kin. So if you disperse all around, I mean, cooperation should be negatively selected if you disperse too much. But if you say, well, okay, let's favor kin cooperation, let's stay together, this is not a suitable solution in the end on very long time, because sometime you will need to disperse. So the solution is stay together, and when the conditions are very hard, send some, I'm sketching, yeah, but send some individuals to do long distance dispersal. Okay, and quite recently, and you see the names, I'm sure you know, Stefan Jacob, okay, Elvier, Bastion, Julien Cotte, Barthes, it's really mixed with Maurice, okay. We showed that this dispersal syndromes, so the fact that strains differ in dispersal and they differ in the traits that are linked to dispersal behavior, it can impact several biological levels up to the meta-ecosystem function. Okay. We did a couple of experience where we wanted to explore the difference between resident and dispersals, okay? So in this kind of experiment, we usually have a design that is in two steps. In the first step, we make these two tubes dispersal system, we let them disperse to basically split the population between the residents and dispersals. And then we use them, okay, and we inject it in communities with other tetramela species. This was a community level experiment. We put either residents, this mouse is, okay, either residents in blue or dispersals. Okay. Then we measured the consequence for biomass accumulation in the whole community. And in a nutshell, you basically see that if you introduce dispersals, the community will produce a lower amount of biomass and will produce it less fast. Okay, so both the biomass growth rate, slope, and the end value, okay, here will be affected. So again, these characteristics within strain characteristics that make dispersals and residents different can have consequences on community functioning. Two examples, I told you that these microcosm systems are good examples to do G by E environment. And two examples, one is an early one, relatively easy. We were interested in understanding density-dependent dynamics, okay? Dispersal, sorry. So, all strains adjust their dispersal rate according to the density. We observed a generally negative density-dependent dispersal. So that's interesting also to know. Often we assume that dispersal is positively dependent to density, because density is seen as a cue for competition. So if local competition is too high, then I should emigrate. But the other is true too. Okay. Yesterday with the students, I used that analogy. if you have to choose two restaurants, one has a very long queue and one has a very short queue. You could say, okay, I'm going to the very short queue because there's competition for time. But you could also have the opposite thinking and say, well, if everyone is going to that restaurant, that one is likely much better. So this is more in that kind of, we don't know mechanistically exactly how it works. But this is a bit more, deep thinking is, well, if there are many people here, it's likely a good habitat. So let's stay here. But what is interesting is that it's not big, but it's significant. There is a G by E component. So this means that the strains differ in their reaction to density. So there is a potential for this reaction norm, this plasticity of dispersal for density, to evolve according to selection pressures. Not in this system because we all maintain the clones to be separate, but in nature, that could be. A second example, and it's more complicated. Now, in conservation biology, a big deal now is to understand how extinction drivers, like climate change, habitat destruction, pollution, whatever, these extinction drivers interact. Because we know more and more that extinction drivers are not simply additive, that often they act synergistically. So 1 + 1 = 3. And these systems allow to test that. So we did the G by E design. So we basically looked at how dispersal related traits, movement traits, and other traits, differ among genotypes, but also according to the concentration of nutrients and to the temperature. We manipulate them independently. And this is just an example. You get the movement path linearity. So here at the top of the graph, here, the individual is going straighter, and here, the individual is doing a more tortuous movement. If you want to escape a locally bad situation, well, you're likely to go straight. What you see here is clearly the individuals increase their linearity, whether when food is lacking, and when temperature is too high or too low. But the most interesting part is that there's also a very large variation between the strains. So there is a G by E. And if you look here at variance partitioning, so I'm speaking about movement speed and linearity here, you get a variance partitioning between the temperature effect in orange, the nutrient effect in yellow, the genotype effect in blue, and then the interactions here. And the interaction part, which is how much strains differ in their effects of temperature and nutrient and the triple interaction, is not negligible at all. By the way, you can also see that some traits react highly to temperature and not to nutrient, and some to the other way. That's a bit out of focus. This is one of the 2 papers I will be discussing with the student this afternoon. Okay. I would say maybe the next player in the game in the early days of the Tetramana was Alexis Chen. Now in Toulouse, he's working on tits, on birds, but at that time he was hired by Jean to work on this microcosm, and he was quite interested in elements that were linked to sociality, and also later on Stefan also went into the game, so about the use of information. So just here a few results on what are the use of information to adjust dispersal decision by Tetramana. So again, we split resident or we let them split between residents and dispersals, and inject them as immigrants into new system, manipulating the number, manipulating different aspects, and then we measure dispersal rate. And here is a summary on what of the parameters of the information that can be brought by immigrants affected the dispersal rate. So if you put immigrants in a higher number, they could carry and bring to the population in which you inject them the information, oh, because they don't know they are in tubes. Huh? So likely they are around populations with high densities because many are coming. Okay, this does not seem to affect the. This mouse is definitely out. So this does not really impact their tendency to disperse. Okay, but three factors brought information and they react. They adapted their dispersal rate accordingly. So what kind of cooperation strategy these immigrants are from? Some strains are more cooperative, some are more selfish, okay? Some information on the landscape matrix characteristics, so the distance of the corridor and things like that, and habitat quality. But more importantly, some of this information also had a genotype-dependent use of information. So some of the strains reacted by, for example, increasing the dispersal rate if they see immigrants with this kind of information, and some had the opposite reaction. So with Stefan, we work quite a lot on habitat choice. So habitat choice is basically, I would say that Stefan is the guy who brought that idea of working on habitat choice in T20. At least, I mean, I'm sure it's not only his ideas, but I work with him on that, but it's more to his credit than mine. Okay, and this we looked at how habitat choice can change the consequence of dispersal for local adaptation. So again, it's very simplified, but dispersal, as I told you, dispersal is expected to negatively affect emergence of cooperation, okay? But also dispersal is assumed to counteract local adaptation, because if you have many new individuals coming, bring gene flow, there's no potential for the local population to adapt, okay? And this is indeed true if dispersal is random. So you don't know, I mean, basically the immigrants are just random individuals among the gene pool. But if the individual decide actively where they want to immigrate, okay, so they perform habitat choice. So the design is quite complex, and I'm not going to explain it here in detail, but the basic idea was to allow in three patch systems, okay, with two different temperatures, we allowed individuals to choose whether they preferred a colder temperature or a hotter temperature, okay? There's always a blue and a red, and there are many because there are controls and so on, okay? So you let them choose, do you prefer the hot or do you prefer the cold? And you take these individuals and you repeatedly inject them into populations that are at a very much hotter temperature. So you start with individuals that are adapted to a normal temperature, the blue one. You boost the temperature quite a lot, okay? So they are not dead directly, otherwise there's no experiments, but they are not especially well adapted. And week after week, you had new individuals, either that had no choice to disperse, or that actively selected, we prefer the blue or we prefer the red. And you look how adaptation occurs or not, you look at the performance. And this is the result. So this is a measure of local adaptation in the control. So each week you have individuals that had no chance of performing dispersal and habitat change, you see a quite flat line. There is absolutely no improvement of local adaptation. But if you had individuals that in that first step decided actively to go towards hotter temperature, for any reason, likely that they are better adapted, you see that adaptation is boosted. Okay, so dispersal can improve local adaptation under habitat choice. And this habitat choice also is linked to the thermal specialization of the genotypes. So you expect that in specialists, so individuals or genotypes that have a relatively narrow range of thermal performance, so okay, that they are very well adapted to a narrow range of temperature, okay, you would expect that they have evolved dispersal with habitat choice. If you're happy only between 28 and 30, you should have developed some mechanism to actively be able to move to a place where it's 20 to 28 to 30, okay? Well, if you're a generalist, well, you should disperse randomly. You're happy with everything. You basically don't care. You don't need to make habitat choice. That's what you expect, okay? So again, that's mainly Stefan's job, the experiment, okay? Again, 3-patch system. So you let them do habitat choice or not. And you correlate this with thermal performance and especially the niche widths, okay? Whether they are specialists or not. And this is what we observed. So on the y-axis, you have a metric of habitat choice. So higher it is, the more habitat choice they do. 0 means they do not do habitat choice. And this, they do negative habitat choice. Okay, we'll come back on that too in a minute. The first prediction, indeed, we observed it. So individual with a narrower thermal niche do positive habitat choice. They actively disperse towards the temperature that is better for their performance. But to our surprise, where we should observe random dispersal, we observe negative habitat choice. So these guys, the generalists, it's not that they disperse everywhere. They actively preferred temperature in which their performance was lower. So, is the generalist, you gave strain that has this kind of relatively large generalized type, okay, a choice of temperature, well, it should go here. Obviously, no, it goes here. Okay, why? This was completely contradictory. And Stefan and Bart Hagemann also at Mollis made a model to try to explain that. And basically, by playing with, so if temperature is stable through time, this kind of result is completely impossible. Okay? Well, they would just be completely crazy and they would die in the end. Okay, but if temperature is not stable through time, if it varies, and in nature it's going to vary all the time, okay? Then this is the proportion of the time where the temperature was on a margin, okay? Quite hotter or quite colder than average. And this is the duration of this hotter or colder temperature. And this gray zone is an area where the model predicts the evolution and the coexistence of two strategies. A specialist of the central temperature, that's what you expect, okay? So you would expect that some individuals evolve specialist strategies. They have a narrow reach center at the mean of the temperature that is occurring, okay? But also a generalist with a negative choice. And this is why It's because if you're a generalist, what is the trade-off of being a generalist? Is that at your optimum, you are not as good competitor than a specialist, okay? So if you want to survive the competition with specialists, you need to exploit the margins at which these specialists are much less performing than you are, okay? So don't compete with the champion at its peak. Compete with the champions at places where they're not good. I mean, you're not just I'm not in football. Now it's rugby. I have absolutely no idea of people. But I mean, you want to win to make a contest with a rugby player, play chess or whatever. Okay, not rugby. That's exactly the same. And this possibility to do habitat choice is limited by habitat fragmentation, because you could say, how do they know that there is this temperature there and there? I mean, we assume that they should go to what is best, but how do they know that information? Okay, so we made an experiment where we simply varied the lengths of these corridors. Okay, and what we see is that at emigration, if you increase the distance, they are less able to perform choice at immigration. Well, they likely have less information on what is at the other end of the corridor. Okay, but no, at immigration, sorry. But at immigration, they are more choosy at immigration because they don't know what is at the end. Okay, and they are less choosy when they get at the end. Okay, so if the journey is longer, you don't know exactly what is at the other hand. So you will be more careful when leaving, and you will be less choosy when arriving. Okay, so take-home messages, and I've been a bit longer than expected, so still not out of time, but okay. So these Toulouse microcosms are a very efficient system to study dispersal. Okay, it's partly artificial, but we can study questions that have impacts, that may have impact on conservation biology. This is still my basic interest, okay? By testing a hypothesis, unraveling mechanisms that, if these simple species are able to perform such kind of complex behavior, well, it's likely butterflies, lions, and whatever are able to do that in nature too. Okay, so it's us understanding what species are able to do. There is a huge intraspecific variation in tetramina in a lot of things. Okay, and also there is a lot of genetic material for evolution for dispersal to evolve. In all what I showed you, there were still a big question marks. So we saw there are dispersal syndromes. These dispersal syndromes have impacts on populations, communities, even environment. Okay. But still, we know we've repeatedly seen that dispersal and residence differ. But we still don't know, it's the chicken and egg case. We still don't know whether it's a cause or consequence. So if you see that dispersers are swimming faster, is it because there is variation in the individuals for any reason? And those that disperse faster decide more to emigrate, okay, that would be a cause, or is simply a consequence because when they arrive in the target patch, the food is there and the competition is much less. Okay, we still don't know. So now with a PhD student of mine and a postdoc of mine, we are now, and this is also collaboration with Stefan on the concepts, but on the techniques I'm doing, we are doing that really ourselves. We are running experiments now with a newly developed, what we call 2D dispersal systems. So you see these two patches with a corridor. We created a flat version with a start population, a corridor, and a target population. This is a laser cut plastic sheet that is embedded into 2 glass plates. But now this allows us to follow cells individually throughout all the dispersal process. And this is really the beginning, but it's going to be really interesting. So we will be able to study what occurs at the very here where they decide or not to immigrate, in the transient phase and also in the immigration. And these are first results that we got last week. So it's really the beginning, but results are already very interesting. So we made short and long corridors. And in a nutshell, you get 3 curves here. The start patch, the corridor in blue, and the target patch, okay? You see that the start patch is a flat line. We control for it because there is time effect and so on, so they adapt, okay? So these values are relative to what the population is in the start patch is doing. Short corridor, long corridor, same here, movement speed and linearity. What we see is that the first guys to enter the corridor and to disperse the corridor go faster and straighter than those, okay? So it's not a random sample. And this effect is much more pronounced when the corridor is longer. Okay? And when they arrive in the target patch, their speed decreases quite a lot. Okay? That's exactly what you would expect if you have a decision to immigrate. Okay? You adjust your movement, and when you immigrate, you also adjust. So this is going to occupy us maybe for months because the possibilities of the system are just incredible. But okay, so that's is. So as I told you several times, this is a big work of many people over the years, both in Louvain and in Moulis. And I mentioned him several times, but okay, the key founding role of that is really Jean Claubert. So you know Jean Claubert because he founded Moulis, but he also created these systems. So, sorry for using 1 hour. I'm always speaking much more than I thought, but I'm happy to answer any question you could have.